I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
For the November Cranberry Virtual Brown Bag, we take a couple of months off for harvest, and then we group back together post-harvest to hear all through the growing season. We've heard a lot from our Wisconsin specialists, but we want to make sure that we're plugged into the rest of the nation and the rest of the cranberry growing world as well. So today we're going to hear from researchers out in British Columbia and in Massachusetts and in New Jersey to make sure that everything that is going on, we're getting to learn about and take advantage of and stay connected with. So we're looking forward. I want everyone to feel comfortable asking questions. As far as asking questions, you can do either the chat, which I'm monitoring and will ask on your behalf. You can also unmute yourself and ask a question directly. And if you want to ask a question anonymously, you can send a chat message that says just to Allison, and I will ask that question anonymously. So lots of ways to interact. I also want to mention that we need to remind there have been lots of tax changes in this year that will impact the next taxes you file. So I was just asked to say, if you have not yet talked to your tax specialist, you're a little bit behind the 8 ball. Please talk to your tax specialist. After those little housekeeping bits, I want to thank Tom of the Growers Association for helping host these. So Tom, if you want to say good morning.
Well, technically, I'll say good afternoon, and welcome to our continuing virtual brown bags. And thanks to Allison and Alex for putting these together. It's a great opportunity for growers to get together and take a little bit of time to learn on topics. So we try to hold these that have relevant topics, and at times that growers can join. I know a number of you are probably considering getting out into the woods or looking at it this weekend, and we wish you well with that. But I just want to say welcome, and we're going to continue to do these types of programs. We did have one thing to make folks aware of. We did start the process for developing the research program for the upcoming year with the research roundtable that was held on Wednesday, and we had very good participation, and I think we had a good session. That leads to the Wisconsin Cranberry Board annual meeting, which will be held, somebody correct me, but I believe it is the 29th of November. And but everybody should have gotten a notice on that, and there is a virtual option, so we welcome you to join or come down to the research station, join us at that. But just welcome everybody, and if you're going hunting, have a safe hunt, and hope you and your family have a great Thanksgiving.
Thank you. Thanks to feedback from the education committee, from growers, and from researchers at Wisconsin, I kind of took a little straw poll of who we really wanted to hear from at these brown bags. And a reminder that if you ever have someone you want to hear from, let me know and I'll do my best to book them in. But we had questions about pollination deficit, which is an area of expertise for Sandra Gillespie at the University of Fraser Valley. We wanted to hear Jeff Neihart is new at the genetics program in New Jersey. And so we wanted to hear what he's looking forward toward doing. And then Giverson Mapombi has been doing a lot of work in fruit color and quality. And fruit color and quality also came up a lot at our research roundtable on Wednesday. So those are important and interesting topics. So I'm hoping everyone is looking forward to hearing them. We're going to go through in that order. But Sandra, if you are ready, I will stop sharing my screen so you can share yours. We're looking forward to learning about pollination deficit, what we can do to manage, and what our options are.
All right, thank you so much. So hello, everybody. Thank you so much for inviting me. I'm so pleased to be here. It's morning for me. I had a little worry about time zones, but it's all worked out just fine. Yeah, so I'm going to talk a little bit about some research I did in 2019 and 2020 on pollination deficit in cranberries here in BC. And I always start with a little introduction to pollination in cranberry, though you all are certainly experts on that. That cranberry production does, of course, rely on pollinators. And here, and I'm sure pretty much everywhere else, that's typically done by commercial honeybees. But there's pretty good evidence that some honeybees are less attracted to cranberry flowers due to the low amount of nectar and the fact that they're not terribly good at getting pollen from those flowers. And of course, cranberry flowers are buzz pollinated, much like blueberry. And this is a behavior that honeybees don't do. So cranberry pollination might benefit from the presence of bumblebees because bumblebees can buzz pollinate, right? And this is a behavior where the bee will grab onto the flower and vibrate its body at a certain frequency, and they shake the pollen out of the anthers like a little salt or pepper shaker. And that makes them more efficient pollinators than honeybees in some crops, including blueberry. Blueberry, certain cultivars really benefit from the presence of wild bees. They're also really great pollinators of things like tomatoes. And of course, cranberry falls under this umbrella as well. And so before you go out and say, well, okay, you need lots of bumblebees on your farm, to me, the first question you have to ask is, do you need more bees? If we add more bees, will you get more fruit? And so the thing to remember in cranberry is that it's not actually known to be limited by pollination, because extra fruit that might be pollinated on that upright tend to be aborted, right? So a plant might make an upright with 6 flowers, and bees will pollinate the first 2 and maybe visit all the rest because you've got awesome pollination. But then the plant says, okay, we're just going to invest resources in the first 2 and the other 4 get aborted. And so those extra flowers were a bed hedging strategy in case of a lack of pollination. And the plant was never going to make more than 2 fruits in that upright anyways. But studies of this fruit abortion strategy that the plants have were done on one cultivar, right? So that was Stevens back in the day. And in blueberry, it's pretty clear that cultivars vary in the way that they interact with pollinators. And we wanted to know if cranberry cultivars might vary in their response to increased pollination, right? So you might have a plant or a cultivar that tends to be resource limited, will only ever make 2 uprights, or you could have a cultivar that's more pollen limited. And if you had more pollination, maybe you would make 3 or 4 fruit per upright, and you could increase fruit set via increased pollination. Right. And furthermore, cultivars may interact with pollinators in different ways. There could be variation in flower number or nectar production that might make certain fields more attractive to both honeybees and bumblebees. Okay, so I set out to accomplish 2 objectives. The first and the big one was to measure cultivar variation in pollination deficit to determine if there are different types that might benefit from increased pollination. And the second was to get some preliminary data on the role of honeybees versus wild bees in mitigating deficit here in British Columbia. And so the way we measure pollination deficit is to look at the difference in fruit set between open pollinated and supplemented flowers, right? And so, and this represents the additional potential yield that could be achieved with more pollination, right? So in Stevens, what we might expect to see is in the control that's being pollinated by just the bees, you have about the same fruit set as you would in the treatment. And if you have a varietal perhaps that would benefit from more pollination, if you give it more pollen, you might wind up with an increase in fruit. So we did this work over 2 summers. We looked at 4 cultivars, Stevens, Demoranville, Mullica, and Haines. This was in 4 different commercial farms across 3 sort of general growing regions here in British Columbia. And so we went out with my students mostly went out and we quantified pollinators. We did some netting and counted honeybees for 15 minutes per varietal, at least 3 times during bloom, and counted honeybees rather than capturing them because there's so gosh darn many. And then we estimated crop characteristics such as bloom density and flower per upright to try to get a sense of differences between those crops that might be important to the bees. And then to measure pollination deficit, we took 50 pairs of uprights per varietal per farm. This was reduced to 40 in 2020 to let us increase the sites and the number of varieties that we were looking at. And these are assigned to one of 2 treatments. The control just receives ambient pollination from bees, and the treatment is supplementally pollinated by hand, which means going in there with a paintbrush and getting some pollen off of the petri dish and painting it onto the anther and the stigma so that that plant has more pollen than it knows what to do with. Okay. So we collected bee data 2 to 4 times per varietal, and hand pollinations were at least 6 times in 2019, and then 4 to 6 times in 2020. It was a really hot spring, and the bloom period was truncated, which gave us some issues getting out there as often as we would like. And then in the fall, prior to harvest, we collected the treatment and control uprights. We counted the number of fruits produced. We weighed each fruit, and we cut them open and counted the seeds, because seed number is a way to estimate pollination service and can be correlated with things like fruit weight and things like that. Okay, so what did we find? Well, what we found was this significant site by treatment interaction affecting fruit number. And what the heck does that mean? Well, if you think back to my earlier graph, our control uprights set fewer fruit than our treatment uprights at Chilliwack and Richmond, so the 2 regions we looked at. And so the treatments generally had higher fruit set across all cultivars, which is evidence that there is pollen limitation in these plants. This pattern was weaker at the Delta field station than at our 2 commercial farms in Chilliwack and Richmond. And year was really important, which farmers obviously know that the year-to-year variation is has a really big impact on yields. Fruit set was overall higher in 2020, regardless of varietal and regardless of site. So 2020 was just a better year for cranberry here in BC. So what about the bees? Well, the strongest factor affecting both honeybee and bumblebee abundance on the farms was location. So it didn't vary between varietals. It didn't vary across the year. It was really important where you were. Chilliwack had the highest abundance of honeybees. This is on the log scale. And 2 of our transects were actually right near where they put the hives. So we just had pretty intense honeybee numbers, fewer in Richmond, and then the lowest at Delta. And for bumblebees, we had actually really abundant bumblebees in Chilliwack and in Delta, and lower numbers in Richmond. Okay, so can we put this all together to start to tease things apart? Well, Chilliwack had high numbers of bumblebees and honeybees. It also had the highest numbers of fruit per stem, but it still showed some degree of pollination deficit. It also lacked Demoranville, so that's our 1 variety that had the fewest fruits per stem. So there's a little bit of confounding information there that I have to try to tease apart. Richmond, on the other hand, had moderate numbers of honeybees and bumblebees, lower fruits per stem, but it includes Demoranville, which has really dense bloom, but not very many flowers per stem. But it had very similar pollination deficits to Chilliwack. So the difference between the control and the treatment is really the same. So what that tells me is it's possible that the difference in fruit set is likely not due to pollinator differences, because Chilliwack had tons of honeybees, Richmond, fewer honeybees, lots of bumblebees in Chilliwack. So there might be other things going on with the crop management that is affecting fruit set. Yeah. Okay. And then field station in Delta had the lowest fruits per stem. It has this field of this low productivity, quote unquote, Stevens is a bit of a joke out here. It's maybe sort of drifted away from the true Stevens variety. So that might affect those numbers, but it had the highest numbers of bumblebees because this field research station is right next to this big natural area, this bog, and there's this wonderful export of wild bees onto the farm. And they had very low pollination deficits, right? So what can we conclude? Well, with both years of data combined, we find no evidence that there's differences between cultivars in pollination deficit. Rather, we see variation between locations being important. And as far as the role of honeybees versus wild bees, variation in pollinator abundance across sites didn't seem to be that associated with variation in pollination deficits, but this really needs further analysis. This is 3 sites. Getting at the wild bee impacts on pollination really needs more of a multi-site type of research, which I haven't quite managed to get done yet. Okay, so general conclusions. None of this is, I think, really good news, that none of the varieties that I looked at were particularly susceptible to pollen limitation, which means that, at least for the pollinators here, or the farmers here, what I tell them is you can just manage pollination generally for all your fields, right? If you have a lot of Demoranville or a lot of Stevens, you can kind of stock bees at the same rate and manage things in the same way. this preliminary data doesn't really tell us whether wild pollinators might reduce pollination deficits. The difference between the Delta farm and other locations where Delta had so few honeybees, but still had that sort of very similar fruit set between the control and the treatment, suggests that maybe bumblebees can do a pretty good job. But there's further analysis that I need to do to dig into this. And then I just like to end with this graphic, kind of illustrating how I think about pollination in crops, and that you have these landscape effects affecting whether you have wild bees on your farm, right? And those landscape effects are going to differ between different regions. The way the landscape here is integrated with farms and natural habitat is different than it is in Wisconsin, is different than it is in Massachusetts. And then that determines whether you have bumblebees on your farms, which are on there interacting with the honeybees, whatever your stocking rate is, which then feeds into pollination that is filtered by the physiology of the plant and how many fruit it's willing to set in the first place, all of which is affected by crop management and perhaps varietal differences, which then eventually gives you your crop yield, right? So what I tried to do is just to start getting at some of these impacts here and what the filter is for fruit abortion in different varieties. And there's of course lots of other questions to answer. Okay, with that, I have to thank my field assistants who did most of the work. And this research was funded by the Investment Agricultural Foundation of BC. And I have to put this spiel in here to say that they're not responsible for me. And with that, I don't know if there's time for questions. I'm happy to wait until the general chat at the end. And I apologize for the helicopter noise. We have a bit of a flooding situation going on here.
We do have time for questions. So if there's about 4 minutes before we're up to Jeff. So if you've got questions, go ahead and put those in. I hope that your flooding gets better. I saw that that was looking pretty dramatic. Yeah,
I'm in high and dry here, but I can't get to work. So it's pretty interesting. Let's put it that way. Yeah, it's tough times for the dairy farmers at the moment. That's what's underwater. So.
So I guess questions that while we wait for questions to come in on the chat, do you have plans for future work to look at what it is about various locations or how placement of bumblebees might interact with pollination, maybe given that they fly a little less far than honeybees do?
Oh no, they'd go I think I would say they go the same distance.
Oh, okay, that's good.
Yeah, they'll go multiple kilometers to get food, so over a mile. Sorry. Yeah, yeah. So they'll fly pretty far, and yeah, I mean the advantage of the honeybees is the quantity, right?
Gotcha.
That you can have them there and they're reliable, and what I think is interesting to know is what the benefits are of having both, right? So what the synergies might be between honeybees and bumblebees. And I would like to get at that. I just need to figure out how to do the best way to do it is to do surveys at multiple farms that vary in maybe the proximity to natural habitat and the number of bumblebees, and to look at visit rates and deficit across those. It's just I need a lot of students to do that project. I'd like to do it though.
I did get a question in the chat, and then Christelle, I'll have you ask your question next. What is the general rate of hives per acre growers use in BC, just to get a comparison of what we do in Wisconsin?
I don't remember off the top of my head. I apologize. I'm fairly new to working in Cranberry. No, totally blanking on that.
That's good. Feel free if you follow up with me later. I can.
Yeah, I can find out. I can certainly find out.
Christelle, I'll let you ask your question, and then I'll have another one in the chat as well. Let's go ahead.
Sure. Hi, Sandra. Thank you so much for your presentation. That was great, and that's interesting work. I would say that from the stocking density, I think it's pretty similar. At least we surveyed growers in Quebec.
There you go. Yeah,
about the same than what we're getting in Wisconsin. So 2 to 3 hives per acre would be my guess. But
that sounds right. I get stuck on. I worked in onion seed production, and they do 10 hives per acre, which is really intense. And I get stuck on that number. I think 2 or 3 has to be too low, but that's because the seed production is a whole other area. Yeah, so 2 to 3 sounds right to me.
Yeah, I think it's pretty standard. But I guess my question, and I know you don't have really an answer for that, but you talk about like the native pollinators and how they may be contributing to that pollination deficit. And I think that you're onto something here. I was wondering, when you were doing your sampling at the different sites, you were not at all recording wild bees.
Oh,
right?
we were. Yeah, sorry. So my students would go out, we counted honeybees and we actually netted the bumblebees so I could get the diversity and look at some other things about the bumblebees I'm interested in. So we have counts and how many bumblebees you saw in 15 minutes. And so
I was thinking about the other bees, right? So I was thinking,
the little guys, we looked
about more like commercial.
Yeah, we looked. So the other wild bees like little elastic blossom and solitary bees, we didn't see any.
Okay,
there's a follow-up question in the chat asking, were these bumblebees in the locations purchased or native?
Native. We don't have, as of yet, bumblebees that we can put out in the field here.
Okay.
Bombus impatiens, which you all probably do use, is not native here, though it has escaped. Now it's invasive. That's a whole other problem I'm dealing with. BioBest is trying to develop something. The species doesn't really matter. They're trying to develop a native bee for use in blueberry and cranberry. What I'm trying to figure out is I'm not convinced farmers need them. So that's what I always want to figure out for farmers is it worth your money to buy these bumblebees if, for example, your location is right near what's this place called Burns Bog in Delta, which is a massive natural habitat that is exporting lots of native bees onto these farms. You might not need it, right? So it depends whether more bees would mitigate that pollination deficit, or if there are other things going on in terms of fertilizer or crop management that might be contributing to those patterns.
Awesome. I think we're going to pass it on to Jeff for the next section, and there will be 10 more minutes of questions for everyone at the full end. But thank you so much, Sandra. I learned a lot in terms of pollination priority, and I hope the growers did as well.
Thank you.
Thank you. I will take a couple of questions specifically for Giverson, since Sandra and Jeff both had a couple of individual questions, but also now is the time to start asking questions from anything that you've heard during the presentation, including questions of your fellow growers. I appreciate everyone sharing. I'm happy to take more questions. I know, it was mentioned that Copert, Heidi says that Copert has developed a bumblebee species that is native to the Western states. So they started commercial sales in 2020. So that's still new. I might butcher the Latin, but Bombus vosnesensky. And then if you go to copertus.com and search VOS, that'll be enough. You don't have to spell the Vosnesensky. And then you can also reach out to Heidi or I believe Andrew for more information. So thank you, Tom and Alex. Obviously, the University of Wisconsin Division of Extension, but then a special thanks to Sandra from Fraser Valley University, to Jeff from the USDA in New Jersey, and then from Giverson from UMass. Thank you guys all for connecting with us today and keeping us abreast of everything new in pollination in our pre-breeding genetics program and in fruit color and quality that's going on throughout the world. Great thanks to everyone.